Today we will be talking about variation at the genetic level. I am Dr. Ranajit from YenepoyaResearchCentre. Before going into the topic as such, we will talk about specific learning objectives that we expect you to learn at the end of this class. So we will first learn the definition of evolution in the modern perspective, examples of recent human evolution, then we will little bit talk about Darwinism, then we will define genetic variation, then we will define how genetic variation arise, for example through mutations, then we will talk about how population genetics, the field as such, came into the place by combining Darwinism with Mendelian theories, then we will talk about allele and genotype frequency, and finally we will end our today's class discussing about Hardy-Weinberg equilibrium. So let's define evolution first. What is evolution? In the classic term, evolution means descent with modification, and this is how Charles Darwin defined evolution. And if you have a question about why we are studying evolution, we first must understand that nothing in biology makes sense without in the light of evolution, as correctly said by Dr. Dobzhansky long time back. Now, in the modern context, evolution is defined as the change in allele frequency in the subsequent generation, and this is the definition we will use in the context of population genetics. Evolution, as you can see, can happen at two levels, both at the micro level and at the macro level. In the micro level, as you can see, those 4 sheep, they are all sheep as you can see, and they look quite similar, but there are striking differences in them. Although they are all sheep, same species, but there are striking differences. This is the evolution occurred at the microscopic level or within population level, below species level. Second diagram, as you can see, mentioned as a macroevolution, where all 4 organisms are completely different. And this is the evolution, when we talk about evolution, this is what we actually think about. But evolution can occur at both the micro as well as macro level. And speciation is the concept that separates the microevolution from the macroevolution. For today's class, we will only focus on microevolution, that is evolution at the population level, that is evolution below species level. Now, just to give you a heads on how every living organism are related to each other, and some of you may be surprised to know that human DNA and banana DNA are 50% the same. So you can call yourself 50% banana actually. So in that way, mouse genome and human genome are 92% similar, and chimpanzee genome and human genome are 98% similar. So you can understand how all living organisms are closely related to each other. Now the question is, why are we studying evolution? Are we still evolving? The answer is yes, of course we are evolving. May not be in the way that macroevolution portrays it, but at the microscopic, at the population genetic level, we are evolving. We will give you some examples. For example, the first example that we will see is something called amylase 1 gene. Amylase 1 is a gene that scores for AMY1, that's the gene's name. Have you heard about amylase? Amylase is an enzyme that helps to digest amylose, the sugar. Populations who live on high starch diet, for example the Asians like East Asians, Japanese, Korean, Chinese, and also most Indians who live on mostly eat rice, they live on the high starch diet. If you have multiple copies of this gene AMY1, then you can easily digest that starch, and the result is you will be less likely to be obese, you will remain thin. So most Japanese and Chinese people remain thin despite being on starch diet. On the contrary, the meat eaters, for example people from Africa and some part of Europe, they have much less copy of this amylase 1 gene. As you can mention here, 14 copies of amylase 1 is there in Japanese compared to only 6 copies among Africans. That differentiate the amylase 1, that's why Africans, if they feed on this starch diet, they will tend to be more obese than an Asian. The next example we will see is wisdom teeth. When we will talk about wisdom tooth, you may be familiar that it's a very painful process. The reason, the first question may come into your head, why do we even have wisdom teeth? We do not need wisdom teeth. The reason is human ancestors, especially human close ancestors like Homo neanderthalis, the Neanderthals had a broader jaw. At that point of time, you could easily accommodate those wisdom tooth. But over time, our jaw has become narrower and smaller. But those two teeth are still there. So somehow they want to come out from your gum. And since they do not have a designated place to come out, they come from inside other tooth or something like that, and that causes a lot of pain. But the good thing is, evolution, as I mentioned, microevolution is working very fast, and it has been observed that the babies who are born after 2000, many of them do not have wisdom tooth. And this data came from UK Biobank, which is the largest data set of biological information, and they have clearly mentioned that people are born right now without this wisdom tooth, which is a classic example of microevolution. Next, we will talk about blue eye color. Blue eye color is another evolution just took place only 10,000 years back. Before 10,000 years, most individuals had either black or brown color. But around 12 to 10,000 years back, the whole world was covered with ice. We call it last ice age, last glacial maxima. At that time, some individuals developed this blue eye somewhere near the Black Sea. Those blue eye color actually helped them to survive in this all white condition. Then you may wonder why all over the world people have different skin color. The different skin color comes through a single gene called SLC24A5. At the beginning, when humans started their journey out of Africa, all humans were dark skinned. After several thousand years, one mutation arose in this gene SLC24A5 somewhere in Middle East, most likely in Iran, and that gave rise to this light skin color. And that light skin color spread all over the world and gave rise to this skin color variation. We cannot start any evolution class without talking about Charles Darwin. So since we are talking about population genetics, we have to start from Charles Darwin. Charles Darwin used to live in England, and he was from a very upper class family, and he was supposed to be a church priest. But before agreeing to become priest, he decided to travel around the world through his famous ship HMS Beagle. So he started his journey from Plymouth, and then he traveled all over the world and came back to Plymouth. While traveling this entire route, he spent a substantial amount of time in Galapagos Island. You may have heard about Galapagos Island in terms of Galapagos finches. And here he studied several organisms including finches, and first time talked about genetic variation. So let's see what Darwin meant. So the first concept of Darwin was over-reproduction. What is over-reproduction? Over-reproduction means if you give opportunity to any organism, they will produce more offspring than actually can survive to maturity. That means most will die without food or without shelter or something like that. Now, each one of us, each organism I'm talking about, has some sort of variation. That means either our characteristics, whether physical or physiological, will differ from each other. Some of those characteristics will actually help us to survive the harsh conditions, while the others won't do that. So some of them will survive, that is called selection. And those who survive will pass on their genes to the next generation, and that is called adaptation. So these four concepts together gave rise to famous Darwin's theory of natural selection. Now, to give you a heads on about how much variation you see in the animal world as well as in human world. So you can see there are several tigers photo over there. They are all same species, Panthera tigris, but you can see so many variations in terms of their skin color, stripe pattern, etc., etc. And the lions that you see on your right side, they are all from the same country, Kenya. But look at their mane. Their mane shows lots of variations despite being from the same country. And who knows more variation than being Indian? If you travel from north to south and east to west, you will see lots and lots of variations. Actually, you will see the entire world of variation if you are in India. So the humans show the highest genetic variation nowhere but in our country. The entire idea of population genetics starts from the concept of very philosophical question, who am I? So if you ever asked about yourself who you are, if you try to answer that, there will be three possible answers. One is the language you speak, the place you are born and brought up, and third is your unique gene pool. Basically, all these three together gives yourself the uniqueness and make yourself different from the rest of the world. To be precise, all humans are almost 99.9% identical, and only 0.1% differences are there that makes us unique. That's why we are basically different but the same, we are same but the different. That difference is basically that 0.1%. Now, how that 0.1% difference arise? That 0.1% genetic variation mostly arise due to genetic mutation. Mutation happens due to lots of different factors. We will see mainly three factors. First one is radiation. Radiations, for example, ultraviolet light. Even if you are not exposed UV all the time, the basal level ultraviolet light is always you are being exposed. Then X-rays are there. Then another important factor in modern times are the chemicals. For example, the processed foods, carcinogenic like cigarettes, and then bidis, and for example lots of tobacco items and all that. Then the cosmetics and the cleaning products that we use on a daily basis, they can also cause lots of somatic level mutations. Then finally, there are many infectious agents. For example, HPV virus, there are many bacteria like Helicobacter, they can also cause mutation. Mutation can happen at the two different levels. One, we call it the mutation at the chromosomal level, and then mutation at the DNA level. If mutation happens at the chromosomal level, we call it chromosomal aberration. And as you can see, there are several types of chromosomal aberrations possible, like chromosomal deletion, some segment of the chromosome get deleted. Then duplication, where some segment of the chromosome, like for example B, is getting duplicated here. Then inversion, where the chromosome gets inverted and their orientation, their organization gets changed. As you can see, instead of ABC, it becomes AED. Then finally, translocation, where some segment of the chromosome gets cut and get pasted in some other chromosome. That is called translocation. Chromosomal aberrations are stronger mutation. It can cause several genetic disorders, like example Down syndrome and all. Then mutation can also happen at the DNA level. When it happens at the DNA level, we mostly call it point mutations. Point mutations are of two types, synonymous and non-synonymous. Synonymous substitutions are those which do not change the protein level. For example, as you can see, the actual code of this DNA codon was ATG, and it used to code for tyrosine in the normal protein. In case of synonymous substitution, that G gets changed to A. When it gets changed to A, the amino acid does not change. It still produces tyrosine. So the protein does not change. Non-synonymous substitution, on the contrary, will change the protein. For example, if there is a mutation in A and it converts to G, the new protein will be histidine. Finally, there is a third type where it can generate a stop codon, and as a result, the entire amino acid chain will be truncated and the protein will be incomplete protein. So these are the various ways the variation can arise at the DNA and the chromosome level. In terms of where and how the mutation is arising, mutation can be of two types: somatic mutations and germline mutations. Germline mutations are heritable. That means the mutations that we inherit from our parents, father and mother. Those mutations will happen in every cell of your body, and obviously half of it will be passed on to your germ cells and passed on to the next generation, and 50% of those two will go to the next generation. Somatic mutations are non-heritable and it can happen in your body cells, and since it happens in your body cells, it is not passed to your germ cells and will not be passed on to the next generation. When we talk about population genetics, we only talk about heritable mutations that can be passed on from one generation to the other. For today's class, we will mostly focus on single nucleotide polymorphisms. Single nucleotide polymorphisms are those mutations which at least occur more than 1% in the population. As you can see, three DNA strands are shown there. Most of the strands are exactly same, except there is one point A, G and T variation is shown. In our genome, there are 14 million SNPs are there across our genome. Okay, and some of them are very specific to certain populations. Now we will enter into the world of population genetics. But before going into the world of population genetics, I would like to emphasize about two famous scientists in biology, Charles Darwin and Gregor Mendel. All of you may have already heard about them, and both of them were living in Europe at the same time, and when they were doing their research, that also quite coincided. But the sad thing is they never knew each other. So although Darwin knew about variation, he did not know how variation occurs. Mendel knew the process of variation, but he did not know the idea of evolution. So they never met. So as a consequence, this afternoon tea, which could have solved the biggest mystery of biology, which is evolution, could have been resolved long back. But ultimately, the synthesis of these two great theories happened, and that gave rise to the field called population genetics. So what is population genetics? Population genetics is basically an amalgamation or synthesis of Mendel and Darwin. So let's see what it is. Darwin knew the traits that passed on directly to the next generation, as I mentioned already. Mendel already determined how they are passed on, and it is actually a matter of chance. The combination of Darwin's natural selection and Mendelian genetics is called the synthetic theory of evolution. Later, it came known as population genetics. And these three gentlemen are the three most famous scientists in terms of population genetics. None of them were actually biologists. All of three of them were mathematicians. And later in their life, they got interest in biology, and they coded the theory of natural selection by Charles Darwin and Mendel's law. Combined together, they gave rise to this neo-Darwinism or the synthetic theory. As I already mentioned about mutations, now let's learn another term called alleles. What are alleles? Alleles are the variations of the same gene. For example, as you can see in those two chromosome loci, they are the same chromosome locus. One is showing purple flower, another is showing white flower. So the gene is for the color, and the variation is either purple or white. Same is for the eye color, hair color, what you name it. One example I have also given is the earlobes. Some individuals have detached earlobe. Most Indians actually have detached earlobes. And detached earlobe is a dominant character, and whether you are homozygous dominant or heterozygous, you will have detached earlobe. On the contrary, some individuals, mostly Europeans, have earlobes that are attached to your skin in the face. Now, what is mutation in terms of population genetics? Crow and Kaimura in 1960s, they developed a model called infinite allele model. What does it mean? It means each new mutation creates a completely new allele. Reversion of mutation is very, very rare, and is so rare that it is essentially non-existent. Any single mutation is as likely to happen as any other single mutation. Let's see what is allele frequency. So allele frequency is in a population of people, percentage of allele. Again, we are considering two alleles per individual that are type of this kind of allele. For example, is given by the notation of PA, P small a, PT, etc., etc. For the simplicity of population genetics, we only consider two allele system. That means either you have capital A or you have small a. And the frequency of capital A is P and frequency of small a is Q. Since we have only two allele and only two options, P plus Q equal to 1. Now, as you can already mentioned, the allele frequency cannot be directly observed. It has to be properly calculated from the genotype frequency. Now, what is genotype frequency? In a population of people, percentage of individuals having a particular genotype is called the genotype frequency. And genotype frequency is given by capital A, capital A, small a, or dominant D, heterozygous H, recessive R. The idea of DHR comes from Mendel's laws. Now with the progression of science, we know all genes are not dominant or recessive, they were somewhere in between, but we have kept the notations to show the connection with Mendelian genetics. We can directly observe genotype frequency from the individuals, and we do not need to do any calculation for this. Under Hardy-Weinberg equilibrium, you can calculate the expected frequency from the allele frequency. We will talk about Hardy-Weinberg frequencies. Suppose there are only two alleles, capital A and small a, with frequencies of small p and small q in a population. As I mentioned, p plus q has to be. Now, if individual has two alleles with random independent picks, that means suppose you have a hat of alleles and you are closing your eyes and picking something. Okay, that's how it is done. In that situation, capital A will be p square, because the first one that you are picking is p, the second one you are picking is p, so p multiplied by p is p square. Capital A small a is 2pq, the first one you are picking p, second one you are picking q, and another option is the first one you are picking q, second one you are picking is p, so there is together it is 2pq. Finally, the small a, the both time from the hat you are picking q, so and q is q square. Under Hardy-Weinberg equilibrium, p square plus 2pq plus q square equal to 1. That means again we have only two alleles, p and q, so as combinations of the genotypes will only be around those two alleles. So we can have p square number of individuals, 2pq number of individuals, and q square number of individuals for a particular genotype, and combining them two will be 100%. So p square plus 2pq plus q square equal to 1. Hardy-Weinberg equilibrium is not without assumption. It is basically the most important null hypothesis in population genetics. Hardy-Weinberg equilibrium simply means no evolution. And when no evolution occurs? When there is no natural selection, no mutation, no genetic migration, population is very, very large and randomly mating. Under that concept only Hardy-Weinberg equilibrium will operate. Let's see the assumptions again. So the organism has to be diploid. Hardy-Weinberg equilibrium cannot be applicable to any haploid or aneuploid, polyploid organism. It has to be diploid organism, and the organism has to be sexually reproducing. That means any asexual organism will not be considered. Randomly mating, that means each individual has equal chance to mate with any other individual in the population. The population has to be really large. There should not be any mutation, any migration, or any natural selection operating in the population. The allele frequency in both sexes should be equal. And finally, non-overlapping generation. It means one generation will only start when the previous generation ends. Hardy-Weinberg equilibrium, as I mentioned, for our simplicity, we only do two alleles, p and q, but you must know it can be equally applicable for 3 or more alleles. In 3 or more alleles like shown here, A1, A2, A3, if you have frequencies of P1, P2, P3, the allele frequencies can be given, genotype frequencies can be given by A1 as P1 square, A1A2 as 2P1 P2, and so on. So it can be applicable for any number of alleles. Now why do we care? Why do we care about Hardy-Weinberg equilibrium for evolution? The first thing is, if generation n, suppose you are in the nth generation, is in Hardy-Weinberg equilibrium, so will be generation n minus 1, the next generation. And the allele frequencies will also remain the same. That means there will be no change. No change means no evolution. So evolution will not occur generation after generation. In order to have any sort of change, you need to get rid of one or more Hardy-Weinberg assumptions. Either you have some sort of mutation or migration or natural selection or some population has to be smaller size, something like that has to happen to change something. That means evolution to occur. To get rid of any bad or deleterious allele, you need natural selection. Natural selection has the only power to get rid of bad alleles. In absence of natural selection, genetic diversity will be maintained by random genetic drift. Now, finally, let's see how we can calculate the disease frequency from the allele frequency. First, we will do recessive calculation. Suppose a recessive disease which is fully penetrant, that means if you have the mutation, you will have the disease, is observed with the frequency of capital R in the population. Now we have to find out the frequency of the disease allele. Since this is a recessive disorder, so it can only arise if both bad copies are present. That means R equal to Q square. So very simple, Q will be square root of R. That means in case of recessive disorders, the recessive allele frequency will be square root of R, which is the disease frequency. If the same thing happens for dominants, things change. Suppose we are talking about a dominant disorder, and as a consequence, since it's a dominant disorder, it will happen in both the homozygous dominant individual as well as the heterozygous individual. Now what is the disease frequency? Now as you can understand, since it's a dominant disorder, so both homozygous dominant as well as the heterozygous individuals can have the disease. So P square plus 2PQ. Now if somebody has dominant disorder, the value P square is extremely small. Just to make a note here, dominant disorders are extremely rare, very rare. As a result, P square is very small compared to 2PQ. Further, if the disease is very rare, Q will be close to 1. That means most individuals in the population will not have the disease. So the disease frequency will remain as only 2P. So as a result, the disease frequency will be, so dominant allele frequency P will be disease frequency divided by 2. Let's summarize what we have learned today. The modern definition of evolution thrusts on the change in allele frequencies across generations. Humans are still evolving across various traits. New alleles are formed through mutations. There are other factors that we will learn in subsequent classes. Darwin's natural selection combined with Mendel's law gave rise to the field of population genetics. Hardy-Weinberg assumptions are only valid when there are no mutation, no migration, natural selection operating in the population, and population is very large and randomly mating. With that, we will finish today's class. We will see you next time.